السلام عليكم ورحمة الله وبركاته. طلاب المرحلة الثانية كلية الصيدلة جامعة بابل. Our topic today is the autonomic nervous system. As you know, the nervous system is divided into central and peripheral nervous system. And we can divide the central nervous system into the brain and the spinal cord. The spinal cord is responsible for the motor action, while the sensory input will pass into the brain. The peripheral nervous system composed of two parts, which are the somatic nervous system or the voluntary action, which is involved the motor and sensory divisions, while the autonomic nervous system is responsible for the involuntary innervation, and it is conclude from the sympathetic and the parasympathetic nervous system. What is the autonomic nervous system and what is its importance for our body? The autonomic nervous system plays a crucial role in the maintenance of our body homeostasis. The autonomic nervous system is largely independent, so it is called autonomous or autonomic, in that its activities are not under direct conscious control of our body. It is concerned primarily with the control and integration of the visceral functions necessary for life, such as cardiac output, blood flow distribution, and digestion. Recently, evidence is accumulating that the autonomic nervous system, especially the vagus nerve, which is a nerve involved in the autonomic nervous system, also influences the immune function and some central nervous system functions, such as seizure discharge. Also, evidence indicates that the autonomic nervous system can also influence the cancer development and progression. The autonomic nervous system is composed of two anatomically and functionally distinct divisions, which are the sympathetic system and the parasympathetic nervous system. Both systems are tonically active. The frequency of discharge of neurons in both systems can either increase or decrease. As a result, tissue activity may be either enhanced or inhibited. There is no sympathetic or parasympathetic action only. There is a homeostasis. There is a frequency discharge of neurons from both sympathetic and parasympathetic systems. This characteristic of the autonomic nervous system improves its ability to more precisely regulate the tissue functions. Without tonic activity, nervous input to a tissue could only increase. So our body needs a tonic activity, which involve the action of both systems, the sympathetic and parasympathetic. Without the tonic activity, the nervous input to the tissue could only increase. Many tissues are innervated by both systems because the sympathetic system and the parasympathetic system typically have opposing effects on a given tissue, increasing the activity of one system while simultaneously decreasing the activity of the other system, result in a very rapid and precise control of tissue function. So this rapid and precise control of tissue function require the activity of both systems. Even they are opposing in their effect, but the increase in the activity of one system will simultaneously decrease the activity of the other system, and this will result in the rapid and precise control of the tissue function. As you see in this figure, we need a homeostasis, which is a dynamic balance between the autonomic nervous system branches. It means the sympathetic and parasympathetic branches. When the parasympathetic system dominated, we are in a condition called rest and digest. This means that the parasympathetic activity is dominated. While when the sympathetic nervous system is dominated, we are in a fight or flight condition, and this condition achieved by the domination of the sympathetic activity. And as you know, sometimes we need to be in rest and digest, but in other situations we need to fight or flight. The sympathetic system controls the fight or flight response. In other words, this system prepares the body for a strenuous physical activity. The events that we would expect to occur within the body to allow this to happen do in fact occur. While the parasympathetic system regulates rest and digest function. In other words, this system controls our basic body function while one is sitting quietly reading a book. As you can see in this figure, these are the branches of the sympathetic and the parasympathetic nervous system. And as you can see, many tissues in the body are innervated by both branches, by the sympathetic and parasympathetic system. While the effect of these branches is differ in the same tissue. As an example, the parasympathetic innervation causes constriction of the pupil, while on the other hand, the sympathetic stimulation causes dilatation of the pupil. Another example, the parasympathetic nervous system stimulates digestive activity. It will increase the gallbladder production and inhibit adrenal production and contract the bladder. This is occur in condition of rest and digest. While when there is a stimulation of the sympathetic nervous system, there will be inhibition of the digestive activity and stimulate the liver to increase glucose release, stimulate the epinephrine and norepinephrine release from the adrenal gland, and relaxation of the bladder to decrease the renal output. Both autonomic and somatic system have an important afferent, which is mean the sensory input. The autonomic nervous system and the somatic system have important afferent or sensory input that provide information regarding the internal and external environment and modify motor output through reflex arc of varying complexity. The sympathetic nervous system is also called thoracolumbar. And why it is called thoracolumbar, we will see. While the parasympathetic is called craniosacral. Okay, the sympathetic called thoracolumbar and the parasympathetic is called craniosacral. The motor neurons in both divisions originate in a nuclei within the central nervous system, which include the brain and the spinal cord. So, the motor neurons originate from a nuclei within the central nervous system and give rise to a preganglionic efferent fibers. A preganglionic efferent fibers that exit from the brain stem and the spinal cord and terminate in a motor ganglia. The sympathetic preganglionic fibers leave the CNS through the thoracic and lumbar, and according to a new information, the sacral spinal nerve. So it is called thoracolumbar. It is the preganglionic fibers originate from the thoracic lumbar area of the spinal nerves, while the parasympathetic preganglionic fibers leave the central nervous system through the cranial nerves, 3rd, 7th, 9th, and 10th cranial nerve. You will understand what we have said in this figure. These are the central nervous system, including the brain and the spinal cord. As you see here, this is the parasympathetic divisions. The motor nuclei is present in the central nervous system, including the brain and the spinal cord. According to this, the parasympathetic division originates from the cranial nerves, in the brainstem, and from the sacral spinal nerves. It is from the brain, from the cranial nerves, and the sacral division of the spinal cord. These are the preganglionic fibers. These are the preganglionic fibers. These are the ganglions. When the fibers originate from the central nervous system, these called the preganglionic fibers, and these are the ganglions, while when the fibers originate from the ganglions, it is called the post-ganglionic fibers. The post-ganglionic fibers, which will pass directly into the target tissue. On the other hand, the sympathetic division of the autonomic nervous system originate from the thoracic, the lumbar part of the spinal cord. It originates from the thoracic and the lumbar part of the spinal cord. As you can see, the preganglionic fibers are very short. While here the preganglionic fibers are long. And as in the parasympathetic division, the sympathetic preganglionic fibers will pass into the ganglions, and then from the ganglions, the post-ganglionic fibers will pass into the target tissue to exert its action. Another figure. This is the medulla of the brain and the brain stem. From there, the fibers originate. These are the preganglionic fibers. This is a parasympathetic division. These are the preganglionic fibers, which are long. This is the ganglion, and then the post-ganglionic fiber, which pass into the target tissue. These are sympathetic, and as we said, they are originated from the thoracic and lumbar division of the spinal cord. These are the preganglionic fibers. These are the ganglions which are present along the spinal cord. These are the ganglions which are present along the spinal cord, and then the post-ganglionic fibers which pass into the target tissue. And as you can see, this is the adrenal medulla, which act as a part of the autonomic nervous system. This is the adrenal medulla, okay, which act as a part of the autonomic nervous system through the release of epinephrine and norepinephrine. Okay. Most thoracic and lumbar parasympathetic preganglionic fibers are short, as we said, and terminate in the ganglia located in the paravertebral chain. And as I said, these ganglia located along the spinal cord, so it is called paravertebral ganglia, and lie on either side of the spinal column. Most of the remaining sympathetic preganglionic fibers are somewhat longer and terminate in prevertebral ganglia, which lie in front of the vertebrae, usually on the ventral surface of the aorta. From the ganglia, what will pass a post-ganglionic sympathetic fiber. As we said, there is a preganglionic fiber, a ganglia, and a post-ganglionic sympathetic fibers which run from the ganglia into the target tissue innervated. Okay, some preganglionic parasympathetic fibers terminate in parasympathetic ganglia located outside the organs innervated, including, this is an exception, okay, and we are talking about the parasympathetic fibers. Some of the preganglionic parasympathetic fibers terminate in parasympathetic ganglia located outside the organs innervated, like the ciliary, the trigo-palatine, submandibular, and the otic ganglia, and we will see it in the next figure. However, majority of parasympathetic preganglionic fiber terminate on the ganglionic cell distributed diffusely or in a network in the wall of the innervated organs. So, majority of the parasympathetic preganglionic fibers terminate on the ganglia, which is situated on the walls of the innervated organ. Several pelvic ganglia are innervated by the sacral preganglionic fibers. Here, what we have said before, this is the parasympathetic division of the autonomic nervous system, which, as we said, is originated in the brain and in the sacral part of the spinal cord. The orange color is the preganglionic fibers, and the other is the post-ganglionic fibers. So, some of the preganglionic fibers, okay, will pass into ganglionic neurons, which are the ciliary, the pterygopalatine, the submandibular, and the otic ganglia, which are responsible for the innervation of the eye muscles, pupils, the nasal glands, tear glands, and salivary glands. The otic is responsible for the carotid salivary gland, and the intramural ganglia, which is the visceral which innervate the visceral organs of the neck, thoracic cavity, and most of the abdominal cavity, while the sacral portion will pass into the intramural ganglia, which innervate the visceral organs in the inferior portion of the abdominal and pelvic cavity. Okay. A large number of afferent fibers run from the periphery to integrating centers, including the enteric plexus in the gut, and the autonomic ganglia and the central nervous system. Many of the sensory pathways that end in the central nervous system terminate in the hypothalamus and the medulla and evoke reflex motor activity that is carried to effector cells by the efferent fibers. Now, part of the autonomic nervous system is the enteric nervous system, which is a large and highly organized collection of neurons located in the wall of the gastrointestinal system. With over 150 million neurons, it is sometimes considered as the third division of autonomic nervous system. It is found in the wall of the GI tract from the esophagus to the distal colon and is involved both motor and secretory activities of the gut. The enteric nervous system is particularly important in the control of motor activity of the colon, and includes the myenteric plexus, which is called the plexus of Erbach, and the submucosal plexus, which is called the plexus of Meissner. These neural network receive a pre-ganglionic fibers from the parasympathetic system and a post-ganglionic sympathetic axons. Again, these neural network receive a pre-ganglionic fibers from the parasympathetic system and a post-ganglionic sympathetic axons. They also receive a sensory input from within the wall of the gut. Fibers from the neuron cells bodies in these plexuses travels forwards, backwards, and in a circular direction. So it will pass forwards, backwards, and in a circular direction to the smooth muscles of the gut to control motility and the secretory cells in the mucosa. As you can see in this figure, this is the mucosa, this is the mucosa, and we pass from the oral to the anal part of the GIT. The submucosal plexus, which contains the secretory neurons, the circular muscles, and then the myenteric plexus, okay? And these are the longitudinal muscles. These are the submucosal plexus and the myenteric plexuses, which involve about 150 million neurons. The enteric nervous system function in a semi-autonomous manner, using input from the motor outflow of the autonomic nervous system for modulation of the GIT activity and sending a sensory information back to the autonomic centers in the CNS. So it will receive input from the motor outflow of the autonomic nervous system and will send a sensory information back into the autonomic centers in the CNS, okay, including the brain and the spinal cord. The enteric nervous system also provides the necessary synchronization of impulses that, for example, ensure forward, not backward, propulsion of the gut contents and relaxation of sphincters when the gut wall contracts. And this is the importance of the enteric nervous system. It will provide the necessary synchronization of impulses that ensure forward, not backward, propulsion of the gut contents and relaxation of the sphincter when the wall of the gut contracts. Okay, this is the synchronization. Now, we have to learn about an important neurotransmitter in the autonomic nervous system, which is called the acetylcholine. And you will study this neurotransmitter and another types of neurotransmitters next year in the pharmacology. Okay, we have to learn some important aspects about this important neurotransmitter. An important traditional classification of autonomic nerves is based on the primary transmitter molecule, which is called the acetylcholine or epinephrine or norepinephrine, released from their terminals and varicosities. A large number of peripheral autonomic nervous system fibers synthesize and release acetylcholine, and they are cholinergic fibers. Why we called these peripheral autonomic nervous system fibers cholinergic fibers? Because they are synthesize and release acetylcholine, and they work by releasing acetylcholine. These includes all preganglionic efferent autonomic fibers and the somatic motor fibers to the skeletal muscles as well. So all the preganglionic efferent autonomic fibers release acetylcholine. Another is the somatic motor fibers to the skeletal muscles also release acetylcholine. Thus, almost all efferent fibers leaving the central nervous system are cholinergic. This is an important note to be in your mind. Almost all efferent fibers leaving the CNS are cholinergic. What does cholinergic mean? Mean these fibers releasing acetylcholine as a neurotransmitter. In addition, most parasympathetic post-ganglionic and some sympathetic post-ganglionic fibers are cholinergic. They release acetylcholine. Okay? This is an important note to be in your mind. All efferent fibers leaving the central nervous system are cholinergic. Most parasympathetic postganglionic fibers are cholinergic. Some sympathetic postganglionic fibers are cholinergic. So, a significant number of parasympathetic postganglionic neurons will not use acetylcholine, but they will use nitric oxide or peptides as the primary transmitter or as a co-transmitters. Okay? A significant number of parasympathetic postganglionic neurons use nitric oxide. You have to pay attention to this figure, which involve the parasympathetic nerves, the sympathetic nerves that originate from the central nervous system. As you can see, this is the central nervous system, including the brain and the spinal cord. This is the parasympathetic nerves, originate as preganglionic neurons, which are long, as we said, and they use the acetylcholine as a neurotransmitter. Okay? And this is the postganglionic neuron, which is also use the acetylcholine as neurotransmitters. Okay, this the N and M can understand its meaning. The N is the nicotinic receptors, the M are the muscarinic receptors, and you will learn about these receptors a lot in the next year in the pharmacology. Okay, so the parasympathetic nerves usually use the acetylcholine either in the preganglionic or in the post-ganglionic neurons. Okay, while the sympathetic nerves, as you remember, they have a small preganglionic neuron. In the sympathetic ganglia, they will use the acetylcholine as a neurotransmitter. This is the post-ganglionic fiber, and this is the target organ. Okay, they use norepinephrine. Okay, they use the norepinephrine in the post-ganglionic neuronal fiber. Okay, some of the sympathetic nerves will use the acetylcholine, and they will use the norepinephrine and the epinephrine. As a neurotransmitters. Most post-ganglionic sympathetic fibers release norepinephrine, as we have seen in the last figure. Most of the post-ganglionic sympathetic fibers release norepinephrine, which is known as noradrenaline. They are noradrenergic, often called adrenergic fibers. That is, they work by releasing norepinephrine or noradrenaline. As noted, some sympathetic fibers also release acetylcholine. Now we have to learn about the dopamine, which is an important transmitter in the central nervous system, and it may be released by some peripheral sympathetic fibers under certain circumstances. So the dopamine is also an important transmitter in the CNS and can be released in the peripheral sympathetic fibers under certain circumstances. Another is the adrenal medullary cells, which are embryologically analogous to post-ganglionic sympathetic neurons. So the adrenal medulla can be regarded as post-ganglionic sympathetic neurons, and it releases a mixture of epinephrine and norepinephrine. Terminals and varicosities of cholinergic neurons, which are the neuronal fibers that release acetylcholine, contain a terminal part and varicosities. These contain a large number of small membrane-bound vesicles concentrated near the portion of the cell membrane facing the synapse, the synapse between the two neural cells, as well as a smaller number of large dense cord vesicles located further from the synaptic membrane. The large vesicles contain a high concentration of peptide co-transmitters, whereas the small clear vesicles contain most of the acetylcholine. What that mean? That means that the acetylcholine is synthesized in the nerve terminals and it is stored in these vesicles. Okay, it is stored in these vesicles. Vesicles may be synthesized in the neuron cell's body and carried to the terminal by the axonal transport. What this means? That these vesicles that contain the acetylcholine are synthesized in the cell body of the neuron, and they are transmitted through the axon into the neuronal terminal. It is transported through the axon into the neuronal terminal. They may also be recycled several times within the terminal after each exocytotic release of the transmitter. Why? Because we need a large amount of the acetylcholine as a neurotransmitter, so we have to recycle it. The neuronal cells will recycle the acetylcholine several times within the terminals after each release of the transmitter. Ultra-fast neuronal firings appear to be supported by rapid recycling of a clathrin-coated vesicle from endosomes in the nerve terminal. Vesicles are provided with vesicle-associated membrane protein. This is important. It is called a vesicle-associated membrane proteins. What is the importance of this protein? Which serve to align them with release sites on the inner neuronal cell membrane and participate in triggering the release of the transmitter. The release site on the inner surface of the nerve terminal membrane contain a synaptosomal nerve-associated proteins. What they do, they interact with the vesicle-associated membrane proteins, okay, and collectively are called fusion proteins. They are important in the fusion, and we will see that in the next figure. This is the axon and this is the nerve terminal, okay? This is the nerve terminal. At this site, this is the vesicle. The vesicle will pass near to the nerve terminal, attached to the cell wall, and it will open, release the acetylcholine. The acetylcholine will attach to its receptors, okay, and the action will provided. So when the acetylcholine is released, it will be recycled. How does it will be recycled? It will be converted into choline and acetate, okay? It will be converted into choline and acetate. So it will pass and reuptake of the choline and acetate into the neural cells, okay, through sodium channels. It will pass into the cell. This is called recycling of acetylcholine, okay. The acetylcholine is synthesized in the cytoplasm from the acetyl coenzyme A and the choline. As you can see, it is synthesized in the cytoplasm from the acetyl coenzyme A and the choline through the catalytic action of enzyme choline esterase. This is the choline esterase enzyme. So the acetylcholine is formed by the acetylcholine esterase and the choline under the effect catalysm of the acetyl transferase enzyme. The acetyl coenzyme A is synthesized in the mitochondria, which are present in a large number in the nerve endings, while the choline is transported from the extracellular fluid into the neuronal terminal by sodium-dependent membrane choline transporters. The acetyl coenzyme A is formed in the mitochondria, while the choline is recycled and passes through the nerve terminal through a sodium-dependent channels. Okay, through a sodium-dependent membrane choline transporter. Once synthesized, I mean the acetylcholine, once it is synthesized, it is transported from the cytoplasm into the vesicle by a vesicle-associated transporter that is driven by proton influx. When it is formed, the acetylcholine will pass from the cytoplasm into the vesicle by the effect of vesicle-associated transporter. This is a vesicle-associated transporter, which will help the acetylcholine to be stored in these vesicles. Okay, the acetylcholine synthesis is a rapid process capable of supporting very high rate of transmitter release. The storage of acetylcholine is accomplished by packaging of quanta of acetylcholine molecule, which contain 1,000 to 50,000 molecules in each vesicle. Okay, most of the vesicular acetylcholine are positively charged quantum amine. These are positively charged. Okay, and is bound to negatively charged vesicular protoglycan. Okay, it is positively charged. Okay, it is positively charged, and it will attach to the negatively charged vesicular protoglycan. The vesicles are concentrated on the inner surface of the nerve terminal facing the synapse, and we have to retain these are the vesicles. They are present here, here, and here in the nerve terminal, which facing the synapse. This is the synapse. Okay. Calcium interacts with the synaptotagmine on the vesicle membrane and trigger the fusion of vesicle membrane with the terminal membrane and opening of the pores into the synapse. I mean the pores of the vesicle. Okay, the opening of the pore and inrush of cation results in release of acetylcholine. Inrush of the cation into the inside of the vesicles will result in the release of the positively charged acetylcholine from the protoglycan and exocytotic expulsion into the synaptic cleft, okay, into the synapse. One depolarization of somatic motor nerve may release several hundred quanta into the synaptic cleft. And one depolarization of autonomic post-ganglionic nerve varicosity or terminal probably release less and release it over a large area. Okay, this is what happened on depolarization of somatic motor nerves and what happened on depolarization of autonomic post-ganglionic nerve. Okay, in addition to acetylcholine, several co-transmitters are released at the same time, but the most important one is the acetylcholine. What happened after the release of acetylcholine? After release from the postsynaptic terminals, acetylcholine molecule bind to and activate acetylcholine receptor, which is called cholinesterase. Eventually and usually very rapidly, all of the acetylcholine release will diffuse within a range of acetylcholine esterase molecule. The acetylcholine esterase very efficiently splits the acetylcholine into choline and esterase. What does that mean? That means that after the release of the acetylcholine molecules, it will bind to acetylcholine receptor and complete its action. Then the acetylcholine will be affected by the acetylcholine esterase enzyme. This acetylcholine esterase will split the acetylcholine into choline and acetate, neither of which has significant transmitter effects. When it is splitted, the acetylcholine, when it is splitted to its basic component, the choline and acetate, it will have no transmitter effect and thereby terminates the action of the transmitter. Okay, most cholinergic synapses are richly supplied with the acetylcholine esterase. The half-life of acetylcholine molecule in the synapse is therefore very short. Why? As you learned that a presence of acetylcholine esterase in the synaptic cleft will split the acetylcholine into choline and acetate, so the half-life of acetylcholine will be very short, a fraction of a second. Acetylcholine esterase is also found in other tissues like red blood cells and other choline esterase with a low specificity for the acetylcholine, including the pterygic choline esterase, pseudocholine esterase are found in the blood plasma, liver, glia, and many other tissues. Returning to the figure, this is the acetylcholine that released from the vesicles, and they will attach to acetylcholine receptors, and in this area there is the acetylcholine esterase present in good concentration. The acetylcholine esterase will split the acetylcholine into choline and acetate, so that is why the acetylcholine half-life of a fraction of seconds. Finally, some figures I think will help you to understand the autonomic nervous system. This is on comparison of the autonomic and somatic motor system, including the sympathetic, the parasympathetic, nervous system, and the somatic nervous system. This is the peripheral nervous system, the neurotransmitters. As you see, the acetylcholine is present in large amount, and the effector organs and the effect that occur on the organs. This is an interesting figure. It shows how chronic stress affects our response. The red line represents sympathetic, and the a blue line represent the parasympathetic stimulation. So the activation of sympathetic and parasympathetic nervous system will occur on a chronic phase when we expose to moderate stress or to a severe stress. This is a moderate stress, and this is a strong sympathetic stress response. As you can see, inadequate proprioception of completion, failure to completely discharge sympathetic arousal. And finally, a chronic stress will leave us in a stressful condition, and we will have failure to recover to the normal sympathetic function. As you see, this is the normal one. This is how the stress increase the sympathetic response, and this is how chronic stress make us fail to recover to normal condition. That's why most of people in our world feel they are in a stressful condition daily. Another figure, body response to stress and anxiety will cause fight or flight reaction. This may sometimes increase colon contraction and result in diarrhea. Some people, when exposed to a stressful condition or into an anxiety, they develop diarrhea due to increased colon contractions, which result in this condition. Okay, and this is how our body responds to a stressful condition. You can imagine that a lion, a bear follow you and you are frightened. What will happen in your body? There will be a hypothalamus activation, which results in sympathetic activation. Okay, there will be an increase in the heart rate, blood pressure, and respiratory rate. The adrenal medulla, which is regarded part of the sympathetic nervous system, will increase the hormones of stress, which are the epinephrine and norepinephrine. This will cause the blood flow to the skeletal muscle and will cause inhibition of digestive activity. Okay, so the sequences of steps in fight or flight behavior, the brain appraises a situation as a threatening and dangerous. So the lower brain structure secrete a stress hormone. The stress hormone signals the adrenal gland to secrete adrenaline, and this causes the muscle to tense, the heart to beat faster, and the liver to send out sugar to be used in the muscles. Okay, another figure show how the anxiety will increase the stress in our body into fight or flight condition. Okay, this is the rest condition, increased sympathetic stimulation in a traumatic event. This is the normal range of sympathetic stimulation. Okay, when there is a traumatic event, it will increase the stress in our body there will be increase in the sympathetic stimulation above the normal range, so we will become in a fight or flight condition. Thank you very much for your attention and wish you all the best. Bye.